Hello, my name is Debbie Sunday, and I'm going to be discussing my research that focuses on natural products for the photodynamic inactivation of bacteria. So first of all, PDI or photodynamic inactivation is a strategy that utilizes a molecule known as a photosensitizer. And this molecule can be activated by certain wavelengths of light and can then go on to participate in two types of transfer reactions, one being electron transfer and the other being energy transfer. And these processes can lead to the production of singlet oxygen species or other reactive molecular species. And these species are very helpful in destroying unwanted pathogens. They can damage microbial cells and ultimately lead to cell death of bacterial cells. Photosensitizers are naturally occurring in many compounds, such as plants, which is one of the reasons why natural products were used for this research. And they often behave as secondary metabolites for defense mechanisms against pathogens and other herbivores in plants. So the ultimate objective of this research is to address the growing concern of antimicrobial resistance, as normal antibiotics and drugs are not as effective in treating patients with infections. And this is a global need that is still containing an unmet solution. So essentially, our objective or our main purpose is to develop a photosynthesizer that can produce an immediate burst of these reactive molecular species that can ultimately overcome antimicrobial resistance and selectively target the highly resistant bacterial cells. So once again, we chose natural products because we had some of the background knowledge that some of these products already contain photosynthesizers, such as Japanese knotweed, and other natural products such as turmeric and aloe vera we chose just to kind of observe what type of activity they would show in our research. So the process in obtaining our samples involves extracting the natural products from their environment, and then we then go on to soak the extracts in ethanol at room temperature, essentially to disrupt the bacterial cell walls or the plant cell walls. After that, we filter and evaporate our solvent, and then we then proceed to go on to various methods of extraction to ultimately maximize our yield. And in the UV-vis spectroscopy graph that you see down below, it essentially just shows the maximum wavelengths that our extracts are absorbing light in. And this is where we are utilizing in our light treatments during our experiments. So to ultimately observe the activities of our natural products, we use two types of methods for our experiments. One is a qualitative method and the other is a quantitative method. The qualitative method is an agar diffusion assay, which involves spreading our selected bacterial strain, which in this case was S. aureus, across an agar plate, and then creating bored holes in that plate where we then add our extracts in. These plates then undergo light treatment, or at least the light plates undergo light treatment, and then after 24 hours of incubation, we observe the zones of inhibition or essentially the area of which our extracts were able to kill the bacteria surrounding it. And then these results are summarized in our table. For our quantitative method, we are using a microbroth dilution assay where we're able to look at our compound and determine the concentration that we need to reduce the cell viability of the bacteria by either 50 or 90%. And as we see here in the heat map shown, green essentially means better activity, while red indicates inactive or no activity. So going forward, we are hoping to look at our most potent extracts, which from these experiments appear to be yellow dog and Japanese knotweed. And then we're going to further use these extracts to test them against more bacterial species, not just the gram-positive, but also the more difficult gram-negative strains, as well as the antibiotic resistant strains. And then from there on, we will observe their photo antimicrobial properties in these strains, and then further on develop our goal of ultimately making a photosensitizer that is able to instantly target these highly resistant bacterial cells. Thank you for listening.